Hi, welcome back. So for this lecture, we're going to be talking about the macromolecules. Now, there's only 4 classes that we really worry about in biology. Those are going to be the carbohydrates, the lipids or fats, nucleotides, and the proteins. Let's get started. So terminology, you're going to have to know a couple of new words. It's nice because these words, like a lot of these macromolecules themselves, break down into some parts. The first word that I need you to know is the word monomer. And so the word monomer, mono means one, and then mer means parts. Okay, we're going to use the word mer quite a few times. So the next one for you to know would be polymer. And if poly means many, then mer, it's made of many parts, right? So that's not so bad. The next one kind of steps it up again. All right, so we got mer in there. Okay. And what else do we have? We have, there's poly. Okay, so polymers. So we're going to do something with polymers. And in this case, we're going to complete a process. So we're going to polymerize, okay? So polymerization means the joining of many individual pieces or groups of pieces together to make something even bigger. So it's the synthesis of something, okay? The next one is to depolymerize. So polymer, we have that in there again, right? So here's poly. Here's, mer. But in this case, we have a D, right? So we're going to depolymerize something. So that means to degrade it. So we're taking something larger and we're going to take it apart or disassemble it into its tinier, smaller parts. All right, so now let's keep working on those prefixes and suffixes and different parts of words that are going to help us just kind of predict what's going on. So now look at these new words. We're just going to add some different things in. So remember we talked about mono already, and we talked about poly, so one and many. But here's a few others. Let's add in this one, di. So that means two, anytime you see that, right? So with these, we're going to talk about the sugars. So sugars are the saccharide. So anytime you see this word here, saccharide, you're dealing with the sugar, right? Or another one would be ose, O-S-E. Okay, we do these a little bit. Hold on, let's get a pen instead of a highlighter, shall we? Okay. So ose as a suffix of anything, that usually means you're dealing with, like glucose or sucrose or fructose or galactose, any of these types of sugars. Okay, so we have single sugar rings, we have 2 sugar rings joined together, and then we have anything more than that is going to fall into the polysaccharide class. All right, the next category is going to be the peptides. So peptides are a little bit different. We're dealing with amino acids. So amino acids are going to be our building blocks, and we'll take a look at what those look like in a little bit. So anytime you see the word peptide, we're dealing with proteins. Peptide, peptide. Okay? So usually the idea of a peptide is a single amino acid. However, it can refer to a chain also. So it can be one or lots. However, the rest of these all have, so di is 2 and poly is many. Again, a dipeptide, 2 amino acids joined together. Polypeptide, more than 2, like many. So let's talk a little bit about molecular synthesis at this point. All right, so we know a few things. We know what a polymer is. That makes sense. Here's 1, 2, 3 joined particles. And then we have an unlinked monomer, right? So mono meaning one, just the one. Okay, that makes sense. So how do we get them to join together? Well, it's important that we know this one. We're going to actually take out the water. So I don't know if you noticed this on the other slide, but we have an OH group. Oops. There it is. We have an OH group on the side and an H on this one. And that's just the right ingredients to make an H2O. So we do what's called a dehydration synthesis reaction. So when we're undergoing dehydration synthesis reactions, we're forming these new bonds by deleting out these little tail groups right here, which are going to be on the ends of every single one of these monomers and polymers that we can use to make even larger chains. Okay, so in this case, because we took out that H2O, we've created a bond now right here on this little spot, right? So here's our former polymer right here, and here's our monomer. And now what we have is an even longer one. So this is how we build up these molecules. So synthesis reactions are key. But now we also have to remember it's not just about building up molecules. Sometimes we have to break them apart, right? Get the energy out, or use them to build other new structures. So in this case, we're going to have to also use water again. This particular reaction is called a hydrolysis, right? So it has hydro, which refers to water, and lysis, okay? And lysis means to break open or split apart. So we're going to use water, and what we're going to do is we're going to replace those missing ends. So on one end, we're going to put an OH group. On the other part, we're just going to stick the hydrogen. So now we used water in two ways. Number one, to build a molecule or synthesize or polymerize a larger molecule, but now we've also used it to break down or degrade a molecule using this hydrolysis reaction. So let's look at this again, and let's look at this with a little more intimidating looking molecule. Now remember, it's just sets of monomers in this case that we're going to join together to make a dimer, right? Two, or a polymer is another way to call it, just more than one. Okay. So let's see here. We're going to work with this little water molecule. Right there. See if I can fix it. More things. There it is. There it is. And that's going to be our target water molecule. Now, if you remember, even though we used a more simplified version a few minutes ago, we're going to still do pretty much the same thing, right? So this is called a dehydration reaction. So dehydration. I swear that says dehydrate. My handwriting is terrible. But anyway, so this is a dehydration reaction. So what we're going to do is we're going to remove the components, making one molecule of water. And what we're going to do is we're going to create one bond now. So now we've gone from two single molecules of glucose, remember O's, that means these are sugars, right? OSE is a big tip-off that most of the time you're dealing with a sugar. And now these two glucose are holding hands, they have a covalent bond here, and they've become a new molecule, a maltose. So after we've been making these molecules, we can actually make them pretty large, and living organisms have been able to put them into some pretty complex shapes and use them for many different things. So for the polysaccharide groups, that's the sugars, we use them in a couple different ways. So plants, I'll put it in plants, so plants are able to use them in two different ways. One is to make starches. So starches are kind of like a short-ish long-term energy storage molecule. So these have very easy to break bonds, right? So we just chain them up and they're ready to burn. However, they also make more complex structures, which are actually used for structural building materials, essentially. So this is cellulose over here. So as you can see, this rigid structure actually has quite a few bonds kind of linking between these covalently bonded strands, all right? And so what happens with this is this will become indigestible parts of the plant, things such as leaves and bark of trees, stuff like that. So for animals, such as this dude right here, right? He's going to have things like glycogen, and another one, which we don't always have, but things like lobsters and cockroaches essentially have chitin. Okay, so glycogen is how we are going to store our sugar a little bit more, sort of long term. So glycogen it tends to branch off a little bit more. Okay, but this is stored within, for example, our muscle tissue. So if we need to all of a sudden run or have a burst of energy, it's there for us, so we can use, our muscle cells can strip it apart, break all these bonds open, and collect the energy. And the other one we were talking about is chitin. And again, we don't really see that as much in something like a human, but that crunchy part of like a lobster shell, for example, that's chitin. It works a lot like cellulose, and that it's indigestible, but it's also a complex carbohydrate. It's a many piece saccharide. Another really important molecule class is the fats. We also call them the lipids. So in animals, at least, we're going to look at how lipids are stored as fatty acid chains attached to a glycerol head. We call these glycerides, right? They're lipids along the line on the glycerol head. Is that helpful? Maybe not. I hope so. But anyway, so here's how it works. So we have these long hydrocarbon chains, right? So these have lots and lots of storage in between all these little bonds of the carbon, right? And hydro, because there's hydrogen there as well, okay? There's also a dehydration synthesis reaction, which attaches these fatty acids to the glycerol heads. This one right here, where we only have one of these fatty acid chains attached to a glycerol head, is called a mono, right, meaning one glyceride. If we look down here, we have 1, 2, 3 attachments. It's called a triglyceride, right? Maybe you get those checked with the doctor sometimes. Yeah. Right, there's also one that I'm not showing you right now. It's just one on the slide, but we're going to talk about lots and lots and lots this semester. It's called a diglyceride, and this particular diglyceride has a phosphate head. Okay, so just keep an eye on that one. It's going to come up later. It's super important, highly abundant, and it's required for all of your cells. Anyway, let's keep looking at these guys. So we have dehydration synthesis, which links our fatty acid chain. Get my marker up. It links our fatty acid chain right here to our glycerol head. And after we've done that, we have either a monoglyceride, a diglyceride, or a triglyceride, 1, 2, 3, depending on how many fatty acid chains are joined. But then we also have something else. We have saturated fats and we have unsaturated fats. You've probably heard about these already. But let's talk about what there are in the molecular level. A saturated fat or an unsaturated fat just refers to how many hydrogens are on a particular carbon chain. So in a saturated molecule, you can see this one is nice and linear and straight. That means that it has the maximum amount of hydrogens that it can possibly carry. That means there are no double bonds between any of these carbons. It's all single bonded. It's saturated with hydrogen, right? So that's why it's a saturated fat. Now inversely, look down here. We have an unsaturated fat. That means that it does not have the maximum amount of hydrogens. It's missing some. And every time we're missing a couple hydrogens, this happens right here, a double bond. And that double bond provides a kink in the chain. See how it kicks off to the side? Well, you can have polyunsaturated fats with lots and lots of kinks in them. So that's what that means. So the next thing is, I know you're thinking, which one of these is good and which one of these is bad? Which is the bad fat, which is the good fat? Well, the truth is, they're both good in moderation and they're both bad outside of moderation, meaning if there's too little or too much in your body. So let's just kind of look at how we can tell the difference just by eyeballing it. Most of us, especially in the first world, tend to get too much of a saturated fat in our diet, right? So saturated fat right here, okay? Oops, pretty, but there it is. So saturated fats tend to be solid at room temperature. It means these chains stack on top of each other. They become things like butter, for example, right? And an unsaturated fat, these are what most people refer to as the good fats, right? So these ones are going to be like aqueous, essentially, at room temperature. Stuff like vegetable oil and olive oil. So that's the two main differences. Saturated fat has a tendency to do stuff like line your arteries and, you know, eventually resulting in atherosclerosis and some pretty rough conditions, leading to things like blood clots and strokes and heart attacks and all that stuff. It's important to remember balance is key. And also, new research is coming out almost every single day that is telling us more about the roles of both of these types of fat in the human diet. Alright, I hope that clears up some questions. So let's move on to the next molecule. So this is a nucleic acid. Basic structure, 3 main parts, okay? So the first part is we have a nitrogenous base, okay? We know it's a nitrogenous base because, look at here, hold on, let me get my highlighter going. Look at all these nitrogens right here, okay? So that's one of the ways we can tell it's a nitrogenous base. Other things that have a lot of nitrogen, amino acids, okay? Interesting thing, that might be a byproduct of some of Earth's early atmospheres. So that means the elements that were available to make some of these essential structures, high abundances of this nitrogen, right? So it's kind of a cool little history lesson there. So nitrogenous bases. Another thing that we look at is this sugar, right? So this, in this case, this is deoxyribose. So we're going to use ribose, right? Just regular ribose. And we're also going to use deoxyribose. So regular ribose, will be for RNA, and then this deoxyribose will be for DNA. Okay? So that means that the molecule that we're looking at right now is a nucleotide of DNA. All right, and the last part is going to be this phosphate group. All right, and this is a group that's, sorry, little frog, a group really ready to react. Look at all these oxygens right here. And you know how oxygen is. Oxygen is definitely an interactor. So that means that we can form bonds or transfer bonds that have lots and lots of energy. This is the basic structure. Sometimes it's shown in different ways. So this is a more molecular type structure, right? It means that we can see things like the different atoms that are there, right? But there are other more simplified versions that we're going to look at like all semester long, okay? And if you use other types of resources, you're definitely going to run into these. So I just wanted you to be used to it. So usually we'll have like the base, and the base is really important. So this is our base up here, okay? So this base is going to tell you which one of, usually about 4, and then there's a special situation with ribose, which we'll get to in a bit, that we're dealing with. In this case, we're dealing with thymine, right? So up here, thymine. So here's a T down here representing that thymine. So notice we don't have all the nitrogens listed in the carbons and the oxygens and everything else, but it's still implied that they're there, right? It's just a simplified version. And then this blue bit over here, sugar, and then over here, this yellow is representing that linkage for phosphate. So let's look at these. These are the bases, some of the different bases that we can have for deoxyribonucleic acid, right? So DNA, okay? This is the stuff that's going to be making up the genome. This is a long-term storage molecule for information, okay? And so remember we already talked about thymine. Here's what it looks like. It's a single circle. Well, there's also adenine, guanine, and cytosine. So there's 4. There's A, T, C, and G, okay? So those are the ones for DNA. All right, so let's keep going. The thing about DNA is you're going to have to know a few things about the bases. So one of the most obvious things, first off, is that some are only 1 circle big, and then some are 2 circles big, or 2 rings big. So that's important because it helps us classify them. So these that are only 1 are pyrimidines. These that are 2 are called the purines, right? So purines and pyrimidines, some of the important things are you're going to need to know which is which. So if I ask you if T or thymine is a purine or a pyrimidine, you're going to need to have the answer for that. So here's one of the ways you can remember. Well, let me start by erasing some stuff. All right. All right. If you guys are enjoying any of that sound in the background, those would be my lovely children. Okay, so here's what I like to do. I like to think of it as pie cut. Because if you have a pie, you're going to need to get yourself a piece. So you're going to need to cut that piece of pie. But spell pie, P-Y, for pyrimidine, right? And then the C and the T are for cytosine, right? And thymine, right here, okay? So that's helpful. The other way we like to look at this one is pure as gold, right? So pure, as in the first part of purine, right? And then A as in adenine, down here, and then G is in guanine, down here. So pie cut, pure as gold. Another thing that's helpful, pyramids, right? So you never see a pyramid that's kind of like a condo, right? You never see them kind of like together in this joined, and then there's like a bridge. You never see that, right? The thing that's important about pyramids is that they're discrete structures. They're single units, right? They stand alone, okay? Right? Here we go. Here's like Gaza. So think of a pyrimidine as being like a pyramid, a standalone ring, as opposed to a purine, which is a double ring, right? Okay, I hope that helps. All right, so this time I want to talk about ribonucleic acid, right, or RNA. So we talked already a lot about deoxyribonucleic acid, so that's the stuff your genome's made out of. But it wasn't the first thing genomes were made out of. Right now, we think that the first genomes on the planet were made out of RNA, right? So it's a slightly different molecule. And when I mean slightly, I mean slightly. I mean, it's pretty much this molecule here, which gives us the R in RNA. So instead of being deoxyribose, right, so if you scooch back to the other slides, you'll see the DNA doesn't have an oxygen here, right? It's just an H. But ribonucleic acid has them both, right? So that's why it's RNA. The other thing about RNA that's important is it has this other pyrimidine that it uses instead of T. Instead of thymine, right? And it has like a slightly modified group. So thymine has a larger group right here, whereas uracil, right, for RNA, much simpler, just CH on that side, okay? So, the thing about RNA too that's important to keep in mind is we still use a lot of it, arguably, and we use it in many different ways. It's multifunctional. So there's going to be three main types of RNA that we talk about in this class. Some that's plugged directly into a ribosome, another that helps transfer amino acids while we're building longer polypeptide chains, and another one, finally, that works as a messenger type RNA. So it gives the instructions for how to build the different types of amino acids we're going to look at. There's also a lot of other things that RNA can do. We'll look at that more in biotech. Things like regulating genes, for example, and even helping us fight viruses. It's cool stuff. Right. So this is another cool one. It's actually the final one we're going to be talking about for this class. So this is called ATP, right? So it means it's adenosine triphosphate. So 1, 2, 3 phosphate groups. See? So ATP is an important molecule because it's an energy transfer molecule. We've been talking about it a little bit already, but this gives you an idea of the structure. So one of the important things is it has an adenine right here. We also have a ribose. And we also have the 3 phosphate groups right here. Okay? We keep that energy bond right, like all that energy we're going to transfer to other molecules here. And typically the one we're going to transfer is going to be this one. Now it doesn't always have to be ATP. Sometimes we have GTP. We can definitely modify it. But the one that we focus on in this class for transferring energy, being the cash money of the cell, and paying enzymes to do work is ATP. All right, so those are the three. So we don't usually just see them as single nucleotides, although that's great. It's not super functional in a biological sense. So what we do see them as are chains. So this is a single DNA strand. They link up, so it's like, so there's phosphate, sugar, phosphate, sugar, phosphate, sugar, right? And so that's how they make this chain. So for DNA, we can have single or double-stranded DNA. For us, for your genome, you have double-stranded DNA. There's also a couple ways it can fold. But for us, in this class, let's just keep an eye on this structure. It's called the double helix. It means that there's 2 strands of DNA. Here's one strand, and here's the other strand. And they're linked together in the middle, right here, by all of our different bases, right? So there's some important things to keep an eye on. All right, hold on, let me clean this up a little bit. Okay, so I gotta draw some more. Also probably should make this a lot bigger, huh? Okay, a little bit better. All right, so now I want you to pay attention to something else. Make this slide bigger. Oh, there it is. Okay, so now there's a way that they always are going to bind, right? So A's are always going to bind to T's, and G's are always going to bind to C's, but what you'll never ever see is you'll never see a C binding to a T, or an A binding to a G, or anything like that. It means that we have specific binding pairs. So always think of this like a GC pair and an AT pair. I think one of the easiest ways to remember it is the only word you can make out of all 4 of those letters is the word at, right? So you know that's one of the base pairs. And so by process of elimination, that means G and C will go together every time. So that's important. And part of the reason is, remember the purines and the pyrimidines again, right? So we had pure as gold. Okay, so it means our purines are going to be A and G. So they are 2 rings big. And our pyrimidines, or pi cut, right, are going to be C and T. So they're only 1 ring big. So now look over here at our model. Okay, so T is going to be 1 ring big, but A is going to be 2 rings big. G is going to be how many rings? 2 rings, right? And C, 1. Okay, how about A again? Oh, 2, and then 1. It means we can only ever collectively have 3 rings across, and it means we need 3. Okay, that's one way to think about it, because it keeps the width of this DNA molecule consistent, right? So we know it's always going to be this far across. That allows it to become a very stable molecule, okay? So hang on, let me adjust this and I'll give you some other ways to think about it. Okay, so here's why that's important to be only 3 rings across. Think of a staircase that you're trying to go up, right? This is a spiral staircase. For us here in New Mexico, remember the spiral staircase in Santa Fe. It's really important that some of these structures are very stable, right? Because again, we only have the handrails that are going up, right? So you don't want to have a staircase that has any situations that look like this, right? So here's like, they wouldn't be too stable. Okay, it would break apart, so it's much better to have a ladder or a staircase that goes like this. So it's really important that we have that 3 rings across rule, right? Okay, I hope that helps them clarify why that is kind of an issue. So remember that, you're going to have base pairing partners. Okay, so always GCs and T's and A's. And you can flip them, like notice down here we have an AT this direction and then a TA in this direction. That's fine, as long as it's just an A and a T that are pairing there. Now, last but not least, I want to go over one last idea with this. So I want you to take a look here in the center. So all of these little, like, there's little dotted lines, right? So those are showing bonding pairs. Okay, we already talked about A's and C's and G's doing that. But I want you to think about it in a different way. So when you look up close to these little, like, disulfide bridges, is what they're called, these little disulfide bridges, there's actually more than just one bond there. In some cases, there's either 2 or 3, and that goes back to the the ATGC thing again. Okay, so remember this. So A's are always going to bind with T's, and G's are always going to bind with C's. And remember, we talked about at being the only word you can really extract out of this, right? So here's something else. It's a 2-letter word, which means there's always only going to be 2 bonds. GCs have 1, 2, 3 bonds between them. Right? So that's another process of elimination type mnemonic, but I hope it's helpful. So essentially, here's what your sequence looks like. When you have a GC, it's going to be 3 bonds. CG, all the way down. Now here's a problem that you might encounter on your homework. Look, it's changed. So I might do something like this, or you might encounter this, where it'll say, instead of having the G's and the C's and the A's and the C's and all this good stuff, you'll just have rings and bonds, right? And so you'll need to use your information that you already know. Things like A and T have 2 bonds, right? And G and C have 3. So here's A and T, and here's G and C. And the other one that's really helpful, remember this one? Right over here. Okay. Pi cut, pure as gold. Okay, so your pyrimidines, right? And a pyramid is a standalone, right? And then purines as gold has to be the other one. All right, so now you have all your information to start decoding the sequence. So let's try it out. Okay, so I have 1, 2, 3 bonds here. So I need to know what my purine is. Okay, my purine looks like it's G. So here's G. So this is going to have to be a C. It's not so bad. Let's do the next one. Okay. So I have 3 bonds again, and my pyrimidines. So the pyrimidines and C. Oh, C. So, oh, well, that's easy. So there's G. Oh, now, 2 bonds. Okay. So 2 bonds brings us back up here to an AT pair. All right. So let's look at our pyrimidine over here. What could it be? Looks like it's a T. All right. And our purine? It's an A. Okay. So now we can kind of flip it. Oh, hey, we know this. This is just a backwards one of these. Okay. And now we're down here again. All right. So what was our purine? Here's a G. And then 1, 2, 3 bonds for C. And then, oh, 2 bonds over here. And let's see. Pyrimidine was T. And then, oh my gosh, you just sequenced based on structure. Brilliant. So this is an amino acid representation. So an amino acid is easy to identify because it has an amine group. So for us, at least for life, biological organisms on Earth, we have about 20 amino acids that we like to use. There can be a lot more than that. Heck, there's been a lot more made on Earth and other planets as well. But we use a system of 20. We link them together to form our polypeptides, which we shape into the different proteins, which are the building blocks, kind of like Legos, of the cell. So there's a bunch of different types of proteins, but let's just group them into 8 different categories to keep things simple for our introduction biology class, right? So there's going to be the proteins that make stuff. We call those structural proteins, right? They're like the rebar and the drywall of all of the parts of the different types of proteins, right? We use them to build things. So there's storage proteins. Now, storage proteins hold things. They're like a reservoir. Transport proteins. They're proteins that take certain molecules from one place to another. Hormonal proteins. We use these to communicate, usually between cells and sometimes within the cell itself. Hormones are really important because they're actually relatively large messengers. Sometimes they work a little slow, and sometimes they can even work over decades. Receptor proteins. Those are the ones that are usually waiting for things like hormones or other cell signal molecules, and then after they receive them, they can execute whatever function they're indicated to. Contractile proteins help us move. Defensive proteins can help protect the cells that we have in our body from foreign invaders or also from other rogue cells that could be dangerous. And finally, we have enzymes. Enzymes is a special class of proteins because they are actually many of these other ones. Lots of contractile ones, structural, transport, receptors, defensive proteins, a lot of these different ones. So it means that they change their shape to execute a function. Oftentimes they use ATP, not always, but a lot of times. So those are really cool. We'll spend some time with those this semester. So let's talk about some of the ways that we can find proteins, or peptides actually. So we can find them in a few different states. And we call these states primary, secondary, tertiary, and quaternary. Primary for one, secondary, tertiary, and quaternary. Right? So that's some notation for you. So here's the differences between each one. They're kind of like steps of a process. Now you can end at any point of the process, and that can be a mature enough molecule, but sometimes we keep going. So when we've translated the sequence from DNA over the type of polypeptide chain that we need to make a specific protein, which is in this single chain, just like a ribbon right here, it's just a sequence. So like valine, glycine, serine, leucine, for example, just 1, 2, 3, 4, and there's nothing else to it, just like a ribbon. We call that primary structure. This is the first structure we see. Now, something that's important about those 20 amino acids, they like to interact. Some are polar, some are nonpolar. And in many other ways, some are acidic, some are more basic. So they're going to interact, repel, or attract each other, and they're going to start creating what we call secondary states. So there's 2 main secondary states that you need to know for this class. Tongue-tied. And the first is the pleated sheet. Okay, think of it as kind of a crumpled accordion type sheet. And the bonds are starting to kind of layer it together so it becomes a little more broad. Another one is the helical structure. So the helical structure is just like a twist, like a little string. Alright, and so you can see here, some of the interactions from the hydrogen bonds are keeping that particular structure together. Those are the two main secondary structures that we'll work with in this class. The next one is going to be tertiary structure. It means the third. So now what's happening are these huge regions, so this is like a very long ribbon, right, that started to fold in on each other. Here's a little area that is a helix. Here's a little area that are the pleated sheets. But it doesn't just stop there. In this case, we have many areas that have pleated sheets. And these pleated sheets have different interactions with the other pleated sheets along the ribbon. And so they continue to fold in on each other. That gives us a tertiary structure. And now is the last one. So this is a single chain, okay? We actually call it a subunit, or a single polypeptide subunit. So after that chain is folded on itself to its maximum folding, to maximize its interactions, it can also globularize or dimerize if it's two or more, polymerize, right, with additional polypeptide subunits to form a quaternary structure. Now these are much larger. You can stop at any point. Most stop between tertiary and quaternary, but those are the four that you need to know. So we learned about the four classes of macromolecules today. We learned about carbs, carbohydrates. We learned just about some of them that are short-term storage energy for energy storage for animals as well as plants. We also learned that some of them are used for making different structures. Then we learned about fats. We learned about how those are arranged and that they're a long-term energy storage molecule. We also learned about nucleotides, how we use them in DNA, RNA, and for that energy transfer in the ATP molecule. And also, finally, we just learned about proteins, how they're amino acid chains, how there's 20 of them that we use in living systems, and how they can fold from primary, secondary, tertiary, and finally quaternary structure. All right, well, I hope that was helpful. Have a great night.